What am I doing? I'm a first year medical student, and in a few days time there is university exam, practical exam. I have the graphs in the exam and I can't identify those graphs. I have not understood anything about those graphs. So what I was doing was just making a small mark behind the graph and making small signs behind each graphs, so that when it comes in the exam, I won't identify the graph from its front end, that is from its characteristics, etc. But I will identify the graph from these small signs that I have made. I can do the same in the histology also. In anatomy, there are histoslides to be observed under the microscope. But I identify the slides from, for example, the slide of liver, it is slightly broken in the corner. From these signs I identify. Hello friends, if this is what is happening to you, that means you have not understood the principles behind these practicals, and then you have to adopt such methods. So in this video, I am going to discuss about the physiology practicals and how you should proceed with the practicals, what are the things that you should understand, you should buy for the practicals, etc. It's in two parts. This one will be experimental and hemat, the other part will be on clinical physiology. And of course, those who find it convenient to be in Hindi, I have another channel which is Hindi, and this same video will also be posted in Hindi. Right, so let's begin. Look, first thing is first principle that I want to tell you as you begin your journey is that you think that if you adopt such methods or cheating or malpractices, teachers would not know. Please don't be under this impression. Teachers have been in this field for long years. They have seen thousands of students, so teachers can easily recognize all such methods. Okay, so if you have such ideas, you can be smarter than the teachers, please drop such ideas. Anyways, let's talk about the business at hand, and that is about the practicals. As you begin and as you enter the physio lab, there will be experimental physiology lab or a section on experimental physiology. This might appear to you to be the most boring part, and maybe rightly so, because what happens is there are certain dark colored graphs, black colored graphs given to you, and they just tell you while giving the instructions that this is the graph of so and so, and these are the things that will be asked in the exam, and you just have to mug it up. It becomes very boring, and these graphs are on amphibian experiments, they are not even on humans. So you might think that we haven't come to medicine for this. We are not becoming doctors for frogs, we are becoming doctors for humans. So why is this being taught to us? Why is this there even in the syllabus? Let me answer this question. Look, animal experiments have been discontinued. I remember my good old days, my first MBBS was way back in 1988. And in those days, we used to actually perform the amphibian experiments. That is, we would dissect the frog, we would take out the tissue, mount the tissue, and actually record the graph on a rotating drum. We have also gone through the scenario of a partly dissected frog jumping here and there in the lab. Yes, we have seen that nazara also. Now, since the animal experiments are not anymore conducted, so that part is gone, then what is the purpose of keeping this part? Okay, there are two important purposes, I would say. One is each graph or experiment is based on some physiological principle. So first thing is that whenever you are learning those graphs, write down at the top of the page what is the principle that you learn in this particular practical or in this particular graph. For example, simple muscle curve. The principle is excitability and contractility. If the graph is of gradation of stimuli, the principle that you learn is recruitment of the motor units. And anyways, I'm going to conduct sessions on practicals where I will also be telling you about these principles. You should write those on your journal, top page of the journal, in these practicals. So that's one thing. Although it's being learnt on the animal, but same principles apply to the humans. And that's the purpose of keeping these practicals. The other purpose is, this is your starting point of understanding how a research is conducted. Eventually, each one of you will have to perform some kind of a research. Either you will go into the research field, or you go for MD, PhD, postdoctoral sciences. That is where you will have to conduct the research. And these graphs and these methodologies is your starting point of understanding how a research is conducted. What are the various levels of research? So therefore, these are still kept in your physiology practicals. If you go to MD, PhD, if you go to pharma industries, research will be the integral part. So this is how you will start learning the research methodologies. Then there will be human experiments. That will be little better. It won't be that boring, but you will perform ergography, spirometry, and you might find that in the laboratory these equipments are little jaded or rusty. Yes, it's quite possible, because there is a lot of wear and tear on those instruments. They are routinely, the practicals are performed on them by the number of students, and therefore, and these are not readily available in the market. If today instrument is, let's say, broken, you can't immediately go to the market next day and just buy. They are custom made. So it will take a long time to buy a new instrument, and therefore you might find instruments in bad shape. Okay, no problem. Try to learn again the principle. For example, ergography, there is a Mozos ergograph where you learn the work done by the forearm muscles. Now this is not done anywhere outside, in the outside world. But there is treadmill ergography, bicycle ergography, which is there in the gym and in the outside world. So you will be learning the principles. If you take up sports medicine as a branch later on, these principles will be handy. You will be well versed with these principles and you can apply them. So that's why human experiments will be interesting. So be attentive, learn those things and perform those things actually during the practical. There will be charts, there will be calculations. You will ask why these calculations are there, why these charts, that's the theory part. No, these charts, these calculations, you will be requiring them even later on, like calculation of dose in pharmacology or calculation of serum osmolarity in medicine. So these things will be required later on in your careers. That's the importance of experimental physiology. The second section will be, or the second part of your practical will be, you will have hematology lab and hemat experiments. This will be little scary part. I'm not scaring you, don't worry. Why scary? Because there will be, these practicals will involve self-pricking. You will prick your finger and collect 1 or 2 drops of blood, your own blood, and conduct the experiments on that. Also, you will find that these methods are obsolete. For example, hemoglobin method, RBC, WBC methods, they are very primitive types of methods. You might think so. But yes, again, you are learning some kind of a principle in each practical. How to collect the blood sample, that's okay, you are collecting on your own, in your own blood sample, you are performing the experiment. But how to match the your sample color with the standard, etc., and what's the normal hemoglobin, you will learn all these principles. So even though if you think that the methods are obsolete, but the principles are very much applicable and very much important to be learned. Coming to the self-pricking part, many of you might think, why do we have to prick ourselves and injure ourselves? That's quite scary. We can't provide ready-made samples to you from the hospital side, because those samples are not checked for all the diseases. So it's not safe to handle those blood samples from the hospital. Safest thing to do is take your own blood sample, handling your own blood sample. That will be the easiest part. Okay, carry your own pen pricker for these experiments. In the diabetology kits, you will get those pen prickers. That's a very easy way of pricking the finger and collecting the blood sample. You wouldn't even know that the finger has been pricked. So remember this always. Apart from that, everything will be provided in the lab itself. Have proper lunch while you are going for the hemat practs. Why I said lunch? Because generally the practicals are in the afternoon sessions, post-lunch sessions. Remember, don't have a very heavy lunch. If you have a very heavy Chinese food or something and go for the practicals, you are going to doze off. You are going to feel sleepy. But a proper, normal, nutritious diet and then go to the lab, because it's quite possible that when you give a painful stimulus to your body, many, or not many, but some people are likely to faint. It's very less in number, this incidence of fainting, maybe 1 in 200 or 250. But it's very much possible, particularly, especially if you have not had proper lunch. If you are going to the practical on empty stomach, then this is a possibility, because a painful stimulus suddenly causes vagus nerve firing and withdrawal of the sympathetic discharge. So suddenly the heart slows down and therefore there is a possibility of fainting. Therefore, a proper nutritious lunch. Anyways, in the previous video, I have told you to take care of your health. So proper nutritious diet will be a part of it. In the coming videos, I will be explaining to you what kind of diet you should have and what things you should avoid. Okay, one more point, do not manipulate the results, particularly in the hematology practicals. There is a tendency that the students tend to manipulate the results because they could not collect the blood properly and they could not perform the practical, so they just write down something. Please avoid this for two reasons. One is, if you manipulate now, then what will you do in the exam? If you are continuously doing manipulations in every practical, in the exams you will fall short because you have never done that practical genuinely. So that's one reason. The other one is, look, this is your starting of you are into the noble profession and this is the start of your journey. So you are going to inculcate some good virtues in you, and this is certainly not a good virtue, manipulation. So cheating, malpractices, manipulations, stay away from these things, because once these things get ingrained in your personality, they will remain forever. So don't do these things from the beginning, at the starting point of your career. All right, so avoid such things and manipulations, etc. An important point that I want to say is, start asking the question why. You are going to ask this question to your tutors, you are going to ask this to your teachers, professors, etc. Why are we doing this practical? What is the importance of this practical in day-to-day life? Or as a future doctor, why do I have to do this and why do I have to know this principle? Start asking these questions, because once you get the answer of why, then the concept is crystal clear in your brain. So you will come to know in a few days in the medical college who is the teacher who is more approachable, and they wouldn't feel offended if you ask the questions. But please maintain certain decorum. Don't ask for the sake of asking. Just ask because you want to learn more. That learning thirst should be seen in you. All right. Finally, one more point. It's about the journal or lab manual. This is the most important part of this video. Complete the journal and get it signed on time. It's very crucial. I told you, Sunday can be kept aside for all these things. You are going to give 2 days for every subject. So 6 days for 3 subjects. Sunday will be kept aside for journal completion, etc. Get it signed on time, because otherwise it will get piled up, and by the end of the year you will make a lot of running around. When there is a time, that time is actually to study for your final exam. You will be completing your journal and running around, running helter and skelter to get it signed from the teachers. They will scold you, they will write incomplete, etc. on your journals. All this can be avoided by just a stitch in time. Saves 9, as they say. So complete it on time. I know many colleges, even I have seen this in very many colleges, that they don't complete it on time and they are scared to go to the teacher, batch teacher, and then the sign is forged. Yeah, in fact, when I was a tutor, I used to sign the journals. Once a student came with this and I was just going through the signatures, and I asked him, this is not my signature. It doesn't seem to be my signature. I have not signed this. Initially, the student was in denial, but then eventually he did accept that, sir, too many signatures and practicals were pending. So, but I said, who has signed this? Who has done this signature? He said, sir, there is one person just outside the campus who is very proficient in making signatures of all the teachers of the college. Wow, take that. And then I said, oh, then that person must be charging. Yes, sir, 2 rupees per signature. These things keep on happening, and we are aware of these things. If you are under the impression that you can outsmart your teachers, please drop this impression right away. You can't. They have seen thousands of students, your teachers. They have seen life, so they do understand what you're doing and what you're going through. So therefore, my discussion continued, actually, that discussion with the student. And when he said 2 rupees per signature, I said, Arey, I am a lenient teacher. You could have come to me. I would have signed myself. I would have done my own signature myself. You didn't have to spend. Then the student cheekily asked, as a joke, sir, how much would you have taken? Okay, that was just a joke, but these things better avoided. Otherwise, in the end, when there is a time to study the theory, you will be right, completing your journal and running around here and there. All right. Do not copy from seniors' journals. I have nothing against seniors. They also have been my students. Okay. So every student is a student for me. But the seniors, once they go to the next year, and they give you advices, it's fine. You can follow the advice. But the journal part, I'll give you an example, one instance. I saw a student's journal, first year student's journal. There was a practical of temperature regulation. And in that practical, somewhere the student had written frog bite. I was surprised. What's this frog bite? Then after a while, I realized, oh, it's a practical of temperature regulation, and the word is frostbite, possibly. You know, the frostbite occurs in extreme cold conditions. The toes, they become ischemic. That's frostbite. But some senior student in some batch wrote frog bite, and everybody copied. Subsequent batches also copying the same thing without realizing how a frog will bite. So it will be a good idea to just ask the teachers what is to be written in this practical and what are the answers to these questions, etc. Finally, for the completion of lab manual or journal, you can take this book. You can purchase this book, V.D. Joshi Sadhana Mendurwar. V.D. Joshi ma'am has been legend of physiology, and the book is extremely popular and accepted everywhere as a universal book. It's a good book for practicals. There is also a prep manual for theory. And Sadhana Mendurwar, her daughter actually, V.D. Joshi ma'am's daughter and my current HOD. And the book is absolutely brilliant. They have given what is required for a first year student. So you can just take this book, gold standard, follow the book. You can write the answers in the journals, and that will be, life will be easier. So these are the practicals that you will be facing in physiology. Next video will be on clinical physiology, because that's a more important part. I have a lot more to say, and therefore I am making another video for clinical physiology.